Okay, well, before I start, I just wanted to give a shout out on the off chance that I misattribute anything or just forget. The whole genome phylogeny is Min Zhao's work. Nor did a lot of the initial processing. I'm going to spend a little time talking about sequence capture, and that was Raphael's wheelhouse. I'm going to give a shout out to Andre about some work he's doing with Rob. And then Sarah Curtis, with some assistance from myself, wrote the read mapping pipeline. So what I'd like to do, as Alice said, is talk about resolving difficult nodes in phylogeny. Of course, we're in the era of phylogenomics, and there are many ways in which you can collect large data sets for phylogenetic analyses. I'm going to highlight two ways that are popular in birds. Most of you know about sequence capture. This is a great method in that it's inexpensive and you can use low quality tissues, but the data types are limited to whatever your probes are, and it's limited to a few thousand loci, which, limited is a little absurd given, when I started, 2 mitochondrial regions was a lot, and 2 to 4 million base pairs of sequence. And the alternative, of course, is just to sequence the whole genome. Here you can generate very large data sets. So I'd like to start out just by showing you Raphael's effort with sequence capture phylogeny. This used 2.5K, so 2,500 locus probe set. Most nodes have strong support. A few problems remain. So the backbone is well resolved, but if I have a support value, that means it's less than 100% support. So few problems remain, but pretty good Manakin phylogeny. 2 genera are non-monophyletic. We expected Neopelma up here, and to some degree we expected Charaxipha plus Antilophia. But what I'd like to highlight is Charaxipha plus Antilophia is one of these low support at the base nodes. So what I'm showing here is some work Raphael did to zoom in on Charaxipha plus Antilophia. I don't want to belabor the details of the methods used, other than to say this is some data filtering plus different methods. Different colors mean different topologies. Your takeaway from this is there's a lot of low support, these numbers, and a lot of different topologies. Almost half of the possible topologies for a rooted tree with 4 groups. We have really 4 groups that come together pretty solidly. So, can we resolve this using whole genome sequencing? As I said, there are a lot of benefits to this. One is many data types. Why does this matter? Well, I'm showing you here some analyses at the base of Neoaves. This is actually a tree of trees. So we're clustering different trees based on different data types. And what I want to highlight is, although the trees are very similar, we have a coding data cluster and a non-coding data cluster. So data type matters. And even when the topology is consistent, you can get differences in branch lengths or support values. The other obvious benefit is large data sets. So what's our goal? Well, we want to resolve 2 genera, I call it in quotes for Charaxipha plus Antilophia, and the genus Lepidothrix, both of which have low support for at least some nodes. A caveat to even whole genome phylogeny is the fact that we could actually have a true polytomy, essentially simultaneous speciation, or we could have a complex phylogeny where we have speciation with gene flow or even hybrid species. What was our specific approach? It was simple. We took tissues, did one library per taxon, sequenced to about 10x coverage, and then we mapped reads. This was the pipeline that we built ourselves that I alluded to, largely Sarah's work. And question one, how much sequence data can we cover from each taxon? So as our test data sets, we used the 259 regions from the Prum et al., or I should say Prum Burv et al. Data set, mostly coding, about 4,600, almost 4,700 UCE regions. Here we use very large flanks, 1 kilobase out from the conserved region. This is more than 10 million bases. And then finally, about 7,000 introns. So again, more than 10 million bases. What I'm showing here is the coverage. In other words, the percent ends, the parts not covered by reads. What you can see is the exons, i.e. the Prum Burv et al. Data set, are recovered best, followed by introns, followed by UCEs. The last of these sort of surprised me because we think of these as, well, ultra conserved. But remember, we're going a kilobase out of the actual conserved region. We see a nice exponential decay in all cases in the uncovered data as we go up in fold coverage based on the amount of sequence. There are sample effects. What you can see here are two taxa that have about 14x coverage, very different recovery. Unfortunately, our friend Kirchsiffia caudata up here was one of our poor recovery samples. Indeed, if we just plot percent ends in one data set versus another, we see a straight line. So now our phylogeny. What we see here is an astral tree based on ML trees for input. Every node has a local posterior of 1.0, except the base of Kirchsiffia plus Antilophia, although I will say that Antilophia is strongly supported here as a sister of Periola plus Lanceolata. And this is pretty good, 0.99 for the introns, a little less for the UCEs. If we instead transition over to a concatenated analysis paradigm, we see a different topology at the base of Kirchsiffia plus Antilophia. This is one that Raphael didn't recover either. So Min got two different topologies. This is one of those cases where concatenation differs from species tree methods. We see another difference down here with Manicus, Pipra, and Macropteryx. In all cases, 100% support. The final thing I'd like to point out is within Lepidophyx coronata, notice these long terminal branches. These samples are as different as other species of Lepidophyx. Andre is doing some work to, with a lot more taxon sampling within Lepidophyx coronata. This is biogeographically sensible. These two down here are Peruvian samples, Panamanian. So we recover the geography. Well, with that, I sort of went through quite fast the phylogeny, but I hope I convinced you that we can get a lot out of low coverage genomics. I want to stress that I didn't show you an exon tree because it'd be a little unfair to compare 250 loci with thousands. We'd like to expand this. And we'd like to move to other regions as well. And we'd like to really examine some complex hypotheses, really get to the base of Kirchsiffia plus Antilophia. Finally, I just want to stress that the Manakin community can use these as a resource. And they're spread across the tree. Obviously, you can't read this, but I am happy to make these slides available to anybody who asks. And with that, I'd like to thank everybody, these NSF-funded projects, and I'd like to thank the Manakin crew for getting me into these wonderful birds. And here's my contact info. Happy to answer any questions if you have them.